Okay, good morning and welcome to this lecture for today. Um, I'm going to be talking about fungi. In this session, I will be looking at the cultural, morphological, biochemical characteristics and how we can identify fungi in the laboratory. Again, I want you to look from the perspective that this is a medical microbiology. Why do I say so? I'll be tilting my attention towards diagnostic mycology. So in most of my examples, in most of my cases, and in most of my procedures, I'll be looking at a situation where we're trying to diagnose a fungal infection in man. I'm saying so because we have botanists here who might be more familiar with fungi in the environment and stuff. But with this knowledge, it's still a comprehensive uh, look at the cultural, morphological, and cultural characteristics of fungi, how we can identify fungi. So I want to start by introducing you to some concept of fungi. The first is mycology. Mycology is a study of fungi. So anytime you come across the word mycology, you should have in mind that they are referring to the study of the different fungi, fungal structures, and growth patterns. Mycosis, as I indicated, is where I'm going to focus on, is the study of fungal infections. We can generally look at it to be fungal infections in animals, including man, but usually it's used when for the fungal infections of man, refer to it as mycosis. Then we have to note that fungi exhibit a dual growth morphology called dimorphism. So I'm now referring to most mycotic fungi. We usually see them grow as a yeast or as a mold. Remember, a yeast is a unicellular structure and a mold is a multicellular structure. So these dimorphic fungi usually exhibit these different characteristics based on their environment and temperature. They are generally guided by the temperature under which they are growing. So if a fungi is growing inside a system, it is likely going to be in a yeast form. That is, if it's a dimorphic fungi. Why, if it is growing on cultural media in the laboratory, usually going to exhibit a mold form, that is a multicellular form. So temperature and the system where the fungi is gradually permit it to exhibit this phenomenon. I've already indicated that molds are filamentous multicellular organisms, while yeast are microscopic unicellular organisms. I'm going to appreciate some of these structures. So this is a yeast grain, and if you do a microscopic stain of a yeast, this is what we'll be observing. This is what we'll observe on the microscopic stain of a yeast. While a mold, as I said, is a multicellular structure that will really grow out of the system. So this is what you really see, a filamentous. I'm sure you guys are familiar with this structure on your bread or on your food. So there are general considerations that we always have to have in mind when we are to study fungi. The first is that unlike bacteria that are greatly classified as either gram-positive or gram-negative based on their peptidoglycan layer, most or almost all fungi are classified as gram-positive. Some are classified as gram-variable. When we look at the nocardia, they are classified as gram-variable, but almost all the fungi that we'll be dealing with, if you do a gram staining procedure on it, you are going to see a purple structure under the microscope. They mostly require oxygen for growth, so almost all fungi except the actinomyces and other bacteria-like fungi are aerobic, so they'll grow in the presence of oxygen. They are eukaryotic organisms. So what do I refer to? Last class, we well identified these to contain membrane-bounded nucleus, and other organelles bounded with membranes like the mitochondria, endoplasmic reticulum, and stuff. So all of them are bounded with membranes. That makes it a true, to possess a true nuclei. Sorry, a true nucleus. So they are heterotrophic in nutrition. Last class, we divided organisms based, we classified organisms based on their mode of nutrition. So fungi are heterotrophic, meaning that they will feed on what already produced organic material. So they'll absorb from their environments. They don't combine carbon dioxide and water to produce organic material. They instead feed on existing organic material, and usually on the dirt and decaying organic material. That's why they are usually referred to as saprophytic. So we have to culture fungi to be able to confirm their presence, to be able to identify their presence. In doing so, which specimen are we likely going to be looking at? In mycosis, usually we'll have fungal infections of the skin and the appendages. These are very common, and to diagnose these infections, we'll need specimen from these areas. So we could take a skin snip, we could take hair, we could take, we could scrape nails and use to culture to be able to confirm the growth and presence of the fungi. Internal fungi, of course, I must know that internal fungal infections are rare and usually common in people with immunocompromised situations like HIV/AIDS and invasive medical procedures. Some of these persons might go down with internal fungal infection, and to confirm that, we'll need sputum. For some cases, we'll need exudates. In some cases, we'll need urine, blood, cerebrospinal fluid, tissue biopsies, and so on. Again, these specimens depend on the clinical manifestation and the suspected infection. When we obtain our specimen, we need to culture on a media, a synthetic artificial media. So there are different media that are available, whether we have general purpose, differential, and more specific. We are going to see some of them shortly. But there are main general considerations that you always have in mind. The first is that we would prefer to culture fungi in a tube. This is a tube. We prefer to culture fungi in a tube, unlike bacteria, where we usually culture on plates. These are petri dish plates. This is a tube. So this is a tube containing a fungi, Cryptococcus neoformans, isolated from, I'm sure, CSF or sputum. So what happens is that when we culture fungi, they grow, and most of them sporulate. And if we grow them on these plates, after about 1 week or 2 weeks of incubation, which is usually the case, when you open these plates, if the fungi was sporulating, you are going to inhale fungal spores. You're going to inhale fungal spores, and that is very, very detrimental to your health. You can acquire the infection. So to avoid that, we prefer to culture fungi on a tube media. We prefer to culture fungi on a tube media. The next reason is that because fungal growth is very slow, unlike bacterial growth that is within hours, fungal growth is within days or weeks. So if we keep it in a plate, there's a high possibility that the moisture content of the media would dry off. So the media will dehydrate. So to prevent this, we'll prefer a tube so that the surface area exposed is smaller, and then the chances of dehydration will also be very, very small. And finally, we culture them for a long time. You can see that keeping them in this place, it requires enormous space, and we might be dealing with more than 100 different specimens. So usually we culture in tubes again to store them easily. So tubes can easily be packaged and stored. So to ensure that we store them easily, we'll culture them in tubes. So there are 3 reasons to summarize. First, to limit the possibility of spore inhalation. Second, to limit the chances of dehydration. And then third, to ease storage. As I said, we culture on different on artificial synthetic media, and different types of media exist depending on the purpose and the type of fungi you are growing. So if you are doing cultivating fungi for studies, or primarily to recover dimorphic fungi or saprophytic fungi from usually the skin,
Good morning.
the media that we typically want to use will include several dextrose agar, which is a classical medium that if you're doing study or research on fungi, usually want to use. This brain hard infusion agar. We also have slants, these on slants, and at times it could be enriched, and it is used when enriched for recovery of histoplasma capsulatum. But when it's not enriched, it could be used for recovery of mostly blood-borne fungi. We have potato flake agar, which is used again for primary recovery of dimorphic fungi. Is somebody trying to ask a question?
Yes, sir.
Go on.
So please, is potato dextrose agar and potato flake agar, are they the same thing?
They are very similar. They are very similar, but I want to differentiate them because uh, I still have potato dextrose agar as one of my media. When I'm talking of similar in terms of composition, but in the same thing, no. They are very, very similar. Okay, we have another group of media, again, directing to which type of fungi we are interested in. If we are interested in dermatophytes, dermatophytic fungi, again, dermatophytic fungi will usually infect the skin, especially the dermis and the lower dermis. So microcell agar is a commercially produced agar that contain the antibiotic chloramphenicol, usually included in the media to inhibit the growth of bacteria, and it also contains chlorhexamide to inhibit saprophytic fungi and some yeast, especially Cryptococcus neoformans. So what are we saying here? We could want to culture fungi from the skin. Fungi from the skin means that it is exposed to so many other contaminants, bacteria, most especially, and other fungi that are not pathogenic. So we want to identify a pathogen implicated in the decay of the skin or the current lesion that we're observing on the skin. So if you don't take particular attention, you are going to have this medium overwhelmed with saprophytes, organisms that are just normal flora and don't cause any pathogenic clinical presentation. So if that is the case, we are going to be unable to identify the particular pathogen. So we need to inhibit their growth so that when we grow, only the pathogen will likely grow. I also want to mention that even the general purpose media I just presented above, potatoes, dextrose agar, and Sabroth dextrose in particular, we could always at one point include chloramphenicol in it, depending on where our sample is coming from. If our sample is coming from the skin, they will always include chloramphenicol in it to inhibit the growth of bacteria. But if our sample is coming from the intestines, usually from blood, for instance, or the CSF and so on, will limit the inclusion of chloramphenicol because this specimen are really considered to be sterile. Yes, Agatha. So what you have noted is that some of the organisms that we want to always eliminate using antibiotics or antifungal will include aspergillus and scoporolus, which are saprophytes and opportunistic pathogens, which will usually not be the cause of our intended investigation. We also want to eliminate Cryptococcus neoformans, which is usually a normal flora and is very pathogenic, but in immunocompromised persons where you can always have meningitis or pneumonia. So, but it's common, as I said, in people with immunocompromised situations. So, if the person is not immunocompromised, we will always want to eliminate it growing because it's always there and but not pathogenic. Bacterial like fungi like actinomycetes are also part of our interest in limiting their growth. So the antibiotic chlorphenicol, while inhibiting the growth of bacteria, will also inhibit the growth of bacteria like fungi. We could also use differential media. Differential media, in this case, we're saying that it permits us to presumptively identify the organism based on its growth characteristics, based on its growth morphology, or its presentation on the growth medium after culture or after incubation. So a good number of media are available for our use in this. We have corn meal agar with a 2080. 2080 is an enrichment substance that is used to permit growth. We also have potato dextrose agar. Again, we can use potato dextrose agar in as a general purpose medium, just like dextrose, sabrose dextrose. Either of them are also very okay for use in cultivation or potato flake, as indicated. But we could as well use potato dextrose agar for demonstration of pigment produced by some organisms. These uh, cultures usually be done on slides in order to induce spore formation. Remember, I said generally we culture bacteria in tubes to prevent inhalation of spores which are produced. But if our intention is to demonstrate spore production as a mode or a means of identifying the pathogen, then we'll have to use a slide and not a tube. Take note about that. So we also have media with special application. I've included all of this in your notes. Sulfuric acid agar for Cryptococcus neoformans, targeting the production of pigmentations. Bird seed agar used to isolate seed neoformans from contaminated cultures. You can read them at your convenience. They are just special media that you could use for more sophisticated investigations. So when we obtain specimen from the body, it could again from the skin, from when we obtain a specimen for investigation, the specimen could be coming from the skin, it could be coming from nail scrapings, it could be coming from internal stuff. But generally, if the specimen is from the skin or nail scrapings, we want to first treat the specimen. Potassium hydroxide, 10 to 20%, but usually in the laboratory we're using 10%. So it is meant to degrade or digest partially the epidermis of the skin, which is very thick. I'm sure if you look at nails or hairs, you see that they are very, very strong proteins, keratin. So we want to digest it partially so that we can go to observe the fungi. So most of the times we'll treat the sample with potassium hydroxide to go to permit visualization of fungi. Okay. After preparing our media, after inoculating our media, we'll need to incubate. So we need to pay attention on the growth requirements of the organism before we can go to achieve sufficient growth within a limited time period. So how do we do that? We need to know the temperature, the best optimal temperature for growth of the organism. A majority of fungi, I must say, which are pathogenic, usually grow at 37 degrees Celsius, while superficial fungi, that is fungi that will infect the skin or the superficial layers of the skin, will grow at 30 degrees Celsius. Remember, the outer temperature is usually lower than the inner temperature. So if a fungi is able to grow at 37 degrees Celsius, be careful. It is a very potential candidate for systemic infections. If a fungi grows at 30 degrees Celsius, know that the fungi is not going to infect the superior layer of the body because it's outer and exposed to lower temperatures. If the fungi is growing at a wide range of temperature from 25 degrees, from 25 degrees from 25 degrees to 37 degrees, remember that 37 degrees is the temperature where a fungi will cause an infection in the body. 25 degrees is environmental temperature. So what does that tell us? The fungi can exhibit dimorphism. It can grow outside as a mold. It can grow inside as a yeast. Good. So we look at the incubation time. How long does the fungi take to grow? It's another aspect that we want to note when cultivating fungi. We must note here that fungi are usually slow growers. They don't grow fast as bacteria. Bacteria usually grow within hours after 24 hours incubation, except maybe Mycobacterium tuberculosis and other few bacteria. You should already be having confluent growth. But for fungi, we want to grow for at least 4 weeks before we can be able to observe substantial growth that can be meaningful. I must also note that there are some that can grow very fast within us. Candida albicans, Aspergillus species, usually grow within 24 to 72 hours, which again is still long compared to bacteria, but very, very short compared to other fungi. Usually, positive culture should obtain from the 7th day. That is, after 1 week of incubation, you start observing if there is growth. Though it takes a long time to grow, we still advise that you monitor your culture. You observe your cultures every other day, especially after the 7th day. Why? Because we want to observe exactly when the first visible growth is noticed and follow it from there. You could allow it for after 10 days and you go, something happened to your cultures, and you won't know after waiting for 10 days before you start the experiment all over. And sometimes, because of desiccation or other environmental conditions, something will go wrong with your incubation procedure. So ensure that you observe your cultures as they are growing. After preparing and manipulating the sample, I want to highlight some techniques that are important for you to get a visible diagnosis depending on your target. For instance, if your fungi is highly motile, if you have a highly motile fungi, you should incubate as an S, as I've indicated here. You as a S, and if it is also rapidly growing, it will be possible for you to see the spreading. Okay, if it is rapidly growing fungi and it is in with other fungi that don't rapidly grow, you are going to find a situation where some are going to max the others. Some are going to max the others. So incubate as an S so that you're able to see rapidly growing fungi, and also if there's some motility to observe, you are also able to see. If you are incubating a slant, the slant should be a straight line. I think I'll have some pictures down to show you. The slant should be a straight line, so you start with a straight line right down, like I'm going to observe. Again, you incubate as there's a lot of distraction in the class, and I'm able to concentrate. So incubation, I've already indicated that if it is a pathogenic fungi, the best incubation period should be around 30 to 37 degrees. If it's a non-pathogenic fungi, the best incubation period should be less than that, around 25 degrees. But of course, that is for botanists. For a clinical scientist, you'll be interested in a fungi only when it is pathogenic. So 30 to 37 degrees will be temperatures that you'll be looking at. Again, note the temperature the oxygen requirements of the pathogen. Is it an aerobic organism or an anaerobic organism? I said earlier that most fungi will require oxygen for growth, except for a very few, especially the actinomycetes. Only the actinomycetes and another limited genera that will require you avoiding oxygen in their environment, but almost all fungi will require oxygen for growth. So make sure that you are incubating under aerobic conditions. So about the general conditions that you have in mind when you want to culture fungi, the first one is, will you need antibiotic in the medium? Remember, antibiotics is when you're collecting specimen from the skin or other exposed surfaces of the body. What is the growth rate of the fungi? Do you start observing after 24 hours? Do you start observing after the seventh day? And finally, note that if it is a fungi that causes systemic infection, the first thing you should know is that you will not need an antibiotic if the sample came from blood or CSF. If the fungi cause systemic infection but can also cause superficial infection, and your samples came from maybe an exudate, then you need an antibiotic because an exudate will contain bacteria and other actinomycetes. The next thing is that systemic fungi will grow at 30 to 37 degrees, while superficial fungi will grow usually around 30 degrees. If it is a non-pathogenic fungi, you should be culturing around 25 degrees. Now, when you culture your fungi, you want to observe the morphology of the fungi as a means of presumptive identification. So you take your plates, you observe your plates. First, you observe the surface topography, whether it is a free-growing fungi, whether it's covering the entire surface of the agar, or it is restricted to a particular angle on the growth medium. You look at the surface texture. Is it a cottony or a woody fungi grain? Is it granular, chalky, velvety, powdery, silky? I have some images to show you about this. The pigmentation of the fungi, does it produce bright color pigmentation, or it doesn't produce any pigmentation at all? Is the pigmentation reverse or obverse? That is it coming from the fungi and is on the fungi, or it is coming from the fungi into the media? So if it's into the media and you're observing the pigmentation from the bottom, we refer to that as reverse pigmentation. But it's coming from the fungi and then on the top on the fungi, then it is obverse pigmentation. Look at the level of the type of mycelia that are formed by the fungi. Is this aerial, or it is growing down into the surface of the system of the medium? So I tried to put together some images here. This is a velvety colony. This is a cottony colony. You see it looking like a wool. This is a powdery colony. You touch it, more touch on it, you see it, shattering on the media, just as you have powder would do. This is a hairy colony on a plate, and this is a granular colony. So after observing with your eyes, we want to confirm the details of what we have observed with a microscope. And with a microscope, we can do further identification. So we can do a test preparation. We can do a slide culture of the organism, and we can look at it using a transparent tape by preparing a transparent tape concentration. I have some images again about this to let you see, have an idea of what a slide culture is like, and so on. You also want to look at the hyphae structure. Is it septate or non-septate? Is the pigmentation demacious or hyaline? So we mean by that, is the demaciation dark or it is transparent? These are things that you want to look at. So this is a slide preparation we are about to do. Down here, you see a typical slide, is a slide tape preparation that you can observe. And here you have the type of pigmentation that we're seeing here. This is a if you look at it, this is the fungal growing here, and you see the pigmentation moving out into the medium, seeping solubly into the medium. These are things that you want to observe and appreciate in identifying the fungi. The hyphae, is it septate or non-septate? So this is a non-septate hyphae. This is a septate hyphae with these branchings that are common in fungi. So if you look at the hyphae again, I said it could either be dimensions with dark pigmentations or highline with transparent pigmentations. Our class will be ending for the first session in 3 minutes time. So as I indicated, I'm trying to do my best to ensure that we have just one recordings. The next session, which I'll start immediately, any recording. So again, after observing the culture under the microscope and identify the pigmentation, we might go further to do biochemical characterization our samples. So the first one is we want to see whether the fungi is able to ferment carbohydrate at its growth. The ability of an organism to ferment carbohydrate is an indication of the fact that it has enzyme systems and subsequently, or as a consequence of the presence of genes in the organism. So if it is able to ferment carbohydrate, we can presumptively identify that. And how do we confirm that? We'll prepare it in a carbohydrate medium and we include what? A pH indicator, a pH indicator in the medium. When fungi grow and ferment carbohydrate, the end product of carbohydrate fermentation will reduce the pH. So we'll have the medium moving from a neutral medium towards the acidic line. In that case, the pH, the bronchosal pH indicator that is included in the system will turn from purple to yellow. So when we grow a medium, our medium, I will come in the next day or after 24 hours, 72 hours, and our medium is now yellow in coloration, which already confirmed that it was able to utilize or it was able to ferment carbohydrate. So we can also do that to confirm whether the organism can assimilate nitrogen and produce gas as well. Gas production as a result of fermentation or assimilation of carbohydrate will usually be observed with through the presence of bubbles in the medium. So we can be able to read some other specific techniques that we want to see if the organism is able to utilize either urea, caffeine, acid, and so on. They are all enumerated here, and you can take your time to go through them. And then finally, we want to stain and observe under the microscope. Staining, again, is a very important tool in microbiology. You want to stain and see if what you're seeing is what is exactly there. So after having your plate, you could stain something in the plate, or alternatively, you could stain samples directly from your skin scrapings or from your tissue. So whenever you stain, you are going to observe a typical, if you do gram staining, you're going to observe a typical purple coloration. If you do different type of stainings that I've outlined here, including the most popular gammaric methenamine silver stain, which is very useful in most skin fungi, you can be able to confirm the presence of the organism. There's also periodic acid skip stain and a good number of others. Please take your time and read. It's very easy to understand. It's a straightforward demonstration. We are ending, the class is ending in less than a minute, so you just have to redial the link and you're back in class for us to discuss more. I'll be able to explain further on these staining procedures during that class, but it will not be recorded since we're trying to minimize the recordings to just one. So I'll meet back in the next few seconds.